Welcome students. We are on to the fourth part of our ongoing lecture, Biotechnology, Principles and Processes. Topic for today's discussion is Processes of Recombinant DNA Technology. We'll talk about isolation of DNA, cutting of DNA and gene amplification using PCR or polymerase chain reaction. Already you have seen the chapter has got 5 learning units. We have already read unit 1 and 2 and 3, and today we will talk about the unit 4, processes of recombinant DNA technology. In the rung of ladders of concept, we are at the 4th rung. So let us first know what is expected of you before we start this lecture. Now you should know the basic tools of recombinant DNA technology. Also should know replication of DNA. You should know the basic differences in nature of cell wall and their mechanism of degradation in different organisms, functioning of restriction enzymes and DNA ligase. At the end of this video lecture, you will be able to explain the process of isolation of DNA from various sources, describe the action of restriction enzymes, explain the steps involved in PCR or polymerase chain reaction, describe the role of thermostable DNA polymerase in PCR. Let's get started. The major processes involved in recombinant DNA technologies are as under. First, we'll isolate the genetic material of the DNA. Then, it is cut at specific location using restriction enzymes. Then, amplification of the gene of interest using PCR or polymerase chain reaction. Then, after creation of recombinant DNA, insertion of recombinant DNA into host cell or the organism, and at last obtaining the foreign gene product. First of all, let us focus on isolation of genetic material or the DNA. First of all, we'll treat the cell from which DNA has to be extracted. If it is bacterial cell, we'll use lysozyme. If it is plant cell, we'll use cellulase to digest the cellulose. And if it is fungal cell, we'll use chitinase. Then after, the cell is treated with ribonuclease so that to remove the RNA. It is done after homogenization or blending. Protein can be removed by treating with proteases. Other molecules can also be removed by appropriate treatment. And this is one of the experiments in your syllabus as well. You can do it very easily using tomato or any other suitable material. Then the content is filtered, and to the filtrate, purified chilled ethanol is added, which precipitates the DNA, which can be seen as fine threads, and it is taken out using a glass rod, and that is called as spooling. This is how we isolate the genetic material. Then cutting the DNA at specific location. The purified DNA molecules is incubated with the restriction enzyme, and the optimal condition is provided for the specific enzyme. Then after, this cut DNAs are separated using gel electrophoresis, and as we know, DNA is negatively charged molecule, hence it moves towards the positive electrode or anode, and the same process is repeated with the vector DNA also, so that you get appropriate DNA molecules. After having cut the source DNA as well as the vector DNA at similar location by the specific restriction enzyme, the cutout gene of interest from the source DNA and cutout vector with spaces are mixed and ligase is added, and we create recombinant DNA. So we have read this in our tools as well, how restriction endonuclease, we have taken example of EcoR1, cuts the vector DNA and the foreign DNA, producing sticky ends, and they are joined to form a recombinant DNA. Now, there was a wonderful technique developed by Kary-Mullis in 1986. If the gene size is found in little amount, it can be amplified. It can be in vitro replicated, right? So PCR is a technique for the in vitro amplification of desired sequence of DNA. What PCR allows us It allows us to produce large quantity of DNA from only few starting copies. And PCR can be used to generate a detectable quantity of DNA from even a single molecule, right? The machine that you can see is a thermocycler. So here we have got specific mechanism and a role of enzymes as well. Let us see them one by one. First of all, we'll just talk about application of PCR. What PCR does? PCR, rapidly amplification of intact genes or gene fragment. It generates large amount of DNA for sequencing, generation of probes specific for uncloned genes by selective amplification of specific segment of complementary DNA, analysis of mutation for medical application, detection of minute amount of DNA for forensic purpose, and amplification of chromosomal regions adjacent to genes of known sequences, and many more. Even you have heard that RT-PCR is done to detect COVID. So it's something that comes to the rescue of mankind ever and ever. What are the steps of PCR or polymerase chain reaction? It is carried out in 3 steps. First step includes denaturation. The double strand, which is to be amplified, is subjected to 95 degrees Celsius temperature, thereby allowing the hydrogen bond to break and the 2 strands to separate. So each separated single-stranded DNA acts as template for DNA synthesis. The second step happens to be annealing, where the temperature in the thermocycler is brought down to 62 degrees, 55 to 62 degrees, and it is kept for about 20 seconds. 2 sets of primers are added, which anneals to the 3' end of each separated strand. So primer acts as initiator for replication. And the third step is extension. The temperature is taken back to 72 degrees Celsius for around 20 degrees, and DNA polymerase extracted from Taq polymerase, thermostable polymerase, that extends the primer by adding nucleotides complementary to template provided in the reaction. So it's very, very important for us to understand, and these steps are repeated many times to obtain several copies of desired DNA. As you can see in the diagram, this region is to be amplified. It is heated, denatured, so that we get single strands. You can see the polarity 5' to 3', and primer is added at 3' end. Why? Because DNA polymerase works only from 5' to 3', and both the sequence acts as point of initiation for DNA polymerase, and DNA polymerase is a special polymerase obtained from Thermus aquaticus, a bacteria which lives in hot sulfur springs, and deoxyribonucleotides are added, thereby allowing the DNA polymerase to complete extension. And after around 30 cycles, we get in tune of 1 billion copies, which is phenomenal. Let us see how it happens. You have seen denaturation taking place. Primers, these are the raw material that happens. Then annealing takes place, and which allows DNA polymerase to extend. So discovery of thermostable DNA polymerase from thermophile bacterium was phenomenal in this regard, and we know that Thermus aquaticus lives in hot spring, and the DNA is resistant to the action of heat. Now, whatever we have read in this unit, the isolation of genetic material using enzymes as cellulase to digest plant cell wall, lysozyme to digest bacterial cell wall, and chitinase to digest fungal cell wall. Cutting of DNA at specific location, we have used restriction enzymes. Then the fragment were separated using gel electrophoresis. Amplification of gene of interest using PCR or polymerase chain reaction. It is in vitro replication of DNA using thermostable Taq polymerase in thermocycler machine. There are three basic steps of PCR: denaturation of DNA strands, annealing with the primer, and adding deoxynucleotide nucleotide, and finally extension using Taq polymerase. There are certain questions for testing your understanding. Let's have a look. During the process of isolation of DNA, chilled ethanol is added to. Options are given: precipitate DNA, break open the cell to release DNA, facilitate action of restriction enzymes, remove proteins such as histones. Which of the following is not correctly matched for the organism and its cell wall degrading enzyme? Algae, methylase; fungi, chitinase; bacteria, lysozyme; plant cell, cellulase. The Taq polymerase enzyme is obtained from name of bacteria given: Bacillus subtilis, Pseudomonas putudia, Thermus aquaticus, and Thiobacillus ferredoxicans. There's one figure being given. It has got three options A, B, and C. You have to point out correct identification that represents what. Look at the options carefully. What is the term used to describe taking out separated DNA from the cell using glass rod? Spooling, spoofing, picking, or DNA spooning? If you have marked your answers, you can check from here. Now some home assignment. Take these questions in your notebook and show it to a mentor. There are some links given for you to visit. Watch the animations. We have read about biotechnology basic principles. We have seen the tools, including restriction enzyme, gel electrophoresis, cloning vector, and ways to make competent host. And in this part, we have read about processes of recombinant DNA technology, isolation of DNA, cutting of DNA, and gene amplification using PCR. In the coming part, we'll be talking about processes of recombinant DNA technology, insertion of recombinant DNA into host, and obtaining foreign gene product. That will be the last part in this series. Watch carefully. This video lecture was presented by Mr. Rajiv Ranjan Singh, PGT Biology, Kendra Vidyalaya, Air Force Station, Kumbhikram, Silchar region. This is a Z at Bhubaneswar presentation. Keep watching. Thank you for viewing.